Hello, are there any questions? Can somebody confirm that you're hearing me?
Yes, I can hear you.
Great. All right, if there's no questions, let's go ahead and begin. Let me share my screen. You should be able to see my screen now. Today is October 11th, and we're supposed to be beginning chapter 4. Please realize that there's going to be a quiz this week, quiz 2, and it'll be covering the end of chapter 2 and chapter 3, as well as lab 3 and lab 4. Excuse me, my voice seems a little hoarse. Any questions about the quiz? If not, let me show you that I've got some grading done. I did grading on 2 labs. Let me see what labs they were. I got lab 3 and 4 graded the first time. I'm afraid I have not graded anything a second time. I also got the last lecture worksheet graded. I think everyone did well on the lecture worksheet. On the last lab, students were having a hard time with it, and I think there was only one student who successfully got 5 out of 5. Everybody else, you will have to redo it if you want 5 points for the lab. All right, you should be seeing my Canvas page now. Let me log into the course. I wanted to point out an announcement. Canvas has stated that they thought they corrected the bug in Canvas, but I noticed that our last worksheet, one student ran into the bug in the discussions. So I'm not sure the bug in Canvas is gone. You might read about it and what you can do if you run into the bug. It does apply to the lecture worksheet. I wanted to point out Canvas College Resource of the Weeks. I tried doing that last time, and it turned out that they changed the format of how they're doing it, and that's why it wasn't working. Last term, which is actually an organized format, it'll look like this. You'll get most of the same information for this term. Like there will be information about the Penguin Pantry, counseling and health services, student success workshops, although this will be workshops for spring term. You'd like to see the workshops for fall term. And then information about tutoring, etc., like information about disability support services. We do have that this term, but it's not organized well. If you click on the link, you have to log into the Clark College website. And I don't know if this is Canvas or what. I think it is. And then you have to scroll down to Resource of the Week. And there will be several links. So this is the Resource of the Week for October 12th through the 19th, Needing Money for College. Resource of the Week for October 6th to 13th, Student Success Workshops. Resource of the Week, 9/22 to 9/30. It doesn't tell you what it's about. Resource of the Week, October 3rd through 7, Emergency Grants. and then September 19th through the 25th. I'm going to click on this because I don't know what One Stop is. And I'm just curious. A resource desk for students and faculty to go and ask questions. Okay. All right, any questions about that? I'm sorry that the link didn't work before. I had a link there, but it went to spring term. And like I said, spring term, it was much better organized. So I've left that link up there in case you want a better organization. Hopefully you're familiar with Resource of the Week. It's just a bunch of resources that you can use at Clark College. And then the other links, I'm not certain that they're going to be terribly interesting. Tutoring services will be useful for you. I doubt travel will be useful for you. Student success coaches could be useful. Student life, you have another link to it, so this isn't a good link to it. Safety committee will probably not be interested in. So most of these other things won't be terribly useful to you. Now, if you're going to form a club, this will be helpful if you're going to organize your club, but unless you're doing that, that link wouldn't be useful to you. All right, any question about any of this stuff?
I do have a question about the last quiz that we took. I was wondering if you've graded the written part of it, the quiz 1A.
I have not graded the recent quiz. Quiz 1A, let me take a look and see if I graded that, because that's not the most recent quiz. I'm going to stop sharing my screen because I'm going to go look into my grade book to look at this. That's just an easy way to look at quiz 1A. Quiz 00A and 00B have been graded.
Because when I look on mine, it says that it's not graded yet.
Well, it's probably not graded then.
Okay.
Quiz 1B is graded. Quiz 1A is not graded. All right, any questions about any of that? Let me get out of here and then I'll share my screen. All right, I think I've already shown this. We're supposed to be doing chapter 4 today. Let me make sure that we have finished chapter 3 because I don't think we did. Can you guys see my screen? I'm going to assume you guys can see my screen.
Yeah, we can see it.
All right. Yeah, we've got a fair amount to do still in chapter 3. We need to talk about the Graham staining and then briefly about a few other stains. So the gram-positive cells stain purple after the procedure. The gram-negative cells stain pink after the procedure. What's the basis for the differential staining? It's related to differences in the cell wall of gram-positive and gram-negative cells. Gram-positive bacteria have a thick peptidoglycan layer, and the crystal violet iodine complex gets stuck in that thick peptidoglycan. The gram-negative bacteria do have a peptidoglycan in their cell wall, if they have a cell wall, but it's a much thinner peptidoglycan layer. It also contains a lipopolysaccharide layer as part of the outer part of the cell wall. So when we look at the cells, let's see if I can blow this up. Here we have a gram-positive cell with the yellow being the cell membrane. And outside of that, we have the purple cell wall. It's thick. It's only showing you 3 layers, but it's many, many times more than 3 layers of peptidoglycan. Okay. The gram-negative cell wall, on the other hand, is thinner. Let me blow this up a little. And you can see that it's got the cell membrane right here, and then a purple cell wall right there containing the peptidoglycan, and then a lipopolysaccharide layer on the outside, which is part of the cell wall. Now it's only showing you 1 layer of peptidoglycan. This is not correct. It's many layers of peptidoglycan in the gram-negative. It's just many layers less than the gram-positive. You got that?
Yes.
And it's much thinner layer of peptidoglycan, and so the crystal violet iodine complex washes out of the gram-negative cells. And then the lipid outer layer of the cell wall that we actually call the outer membrane of the cell wall, it gets destroyed by the decolorizing agent. The decolorizing agent usually has alcohol or acetone or acids in it, and that destroys the lipid outer layer of the cell wall. We never talked about that. So the crystal violet iodine complex is washed out of the gram-negative cells because the peptidoglycan is thinner, and the alcohol decolorizing step removes the lipopolysaccharide layer. That's the outer membrane of the cell wall. The cells will then be colorless as the crystal violet iodine washes out. The gram-positive cells retain the crystal violet iodine because it's a thicker peptidoglycan layer, and the cells retain the purple color. And here we're looking at the gram stain, gram-positive cells, gram-negative cells, the pink ones. When we're looking at the gram stain, you'll often have problems, such as this picture here, where what the heck is that dark stuff there and there and there? And how do you read this slide? What you do is you ignore the bad regions of the slide and focus on the good regions. These bad regions have crystals of crystal violet precipitating on the slide, meaning the crystal violet in solution can precipitate out of solution, forming a crystal. And then when you're using that bottle of crystal violet, you might get that crystal on your slide. Usually what happens if there's a crystal of crystal violet, let me see if I can show that. Yeah, right here. These cells are darker right here than those cells there. And that's because the crystal violet iodine complex is coming out of this crystal and making these cells darker. Sometimes they will look gram positive, whereas when you look at the good regions of the slide, it looks gram negative. And you just ignore these bad regions. Any question about any of that? It's only one problem with the gram stain. You also will have problems if you don't use freshly grown bacteria. The gram stain only works well on young, freshly grown bacteria. If the bacteria is older than 2 days old, you should not use those bacteria, and you should regrow the bacteria to get young cells. And then the gram stain is not universally applicable. Some bacteria cells stain poorly or not at all. That's the mycobacteria. So the results are not always as clear-cut as theory suggests. And sometimes you'll be pulling out your hair because the gram stain, it takes a little bit of practice to do. There is a structure difference between the gram positive and the gram negative cells that not only affects the staining in the gram stain, but also how well they respond to certain antibiotics. For example, gram positive bacteria tend to be killed easily by penicillin G, by cephalosporin, and many detergents. The gram negative bacteria are much more resistant to these antimicrobials. It's because the gram negative bacteria have an outer membrane, the lipopolysaccharide layer, and penicillin G and cephalosporin and some detergents cannot get through this outer membrane of the cell wall. Any questions about any of that? Therefore, penicillin G does not work on gram negative cells. It does work very well on gram positive cells. All right, let's briefly talk about special stains. You're not going to be examined on this, but I want to talk about it because you may use the capsule stain for when you're doing your unknown project. The capsule stain stains the capsules, or really it stains the cells and then the background of the slide, but not the capsule. So right there, this one would be better. You can easily see the cell right there in dark purple, and the background's pretty dark purple too. And then the capsule does not stain. India ink is my favorite stain to work for the capsule stains because it's so simple to use. It lightly stains the cells and then stains the background very darkly, but doesn't stain the capsules at all. Remember, when we're talking about the glycocalyx, you can see a capsule under the light microscope. You cannot see a slime layer under the light microscope, even with the capsule stain. All right, any question about any of that? If not, that's it for chapter 3. Let's go on to chapter 4. Chapter 4 is the functional anatomy of prokaryotic and eukaryotic cells. We will mainly talk about the anatomy of prokaryotic cells because You should have talked about eukaryotic cells in your previous biology class, Biology 160. And so if we run out of time, I will not talk about the anatomy of eukaryotic cells. I will, however, talk about taxis of cells and the endosymbiotic theory, which actually does refer to eukaryotic cells. And then I'll talk about prokaryotic cells. So we'll at least talk about that. If we have time, we'll talk about the anatomy of eukaryotic cells. As is normal, I give you an introductory slide listing the major goals for this lesson and a rough outline. Please know the terms of this lesson. To find all of the objectives you should know, you should look in the chapter, what is it called? Outline and notes. Overview and objectives. No, that's not it either. Objectives, and then the chapter 4 notes. The objectives will be in the objectives document. We are going to talk about how gram-positive and gram-negative cell walls differ, and we'll describe the importance of taxis in bacteria. Any questions about what we're covering? If not, this slide is showing you some differences between prokaryotes and eukaryotes. Prokaryotes is the Greek word meaning pre-nucleus. Literally, it would be pre-kernel, but the kernel is referring to a nucleus. And eukaryote is Greek for true kernel or true nucleus. Prokaryote cells, particularly the bacteria, have one circular chromosome, meaning the chromosome is in a circle and the beginning place is the ending place. The chromosome is not in a nucleus. In eukaryotes, we have linear chromosomes, meaning like on the X chromosome, we have a beginning place and an ending place, and they're not the same place. And then the chromosomes are found in the nucleus. DNA is not associated with histone proteins in bacteria. Histone proteins are a major protein associated with the chromosomes or the DNA of eukaryotic cells. The chromosome of a eukaryote, well, and of prokaryotes, is DNA and protein. And in eukaryotes, that main protein is a histone. Prokaryotes have no organelles. By organelle, I mean membrane-bound organelle. Eukaryotes have membrane-bound organelles. Prokaryotes have peptidoglycan in their cell walls. We're talking about bacteria only. And eukaryotes have no peptidoglycan in their cell walls or in their cells. Prokaryotes divide by binary fission, meaning the cell, when it gets ready to divide, it literally splits into two. Eukaryotes, their cells divide by mitosis. Any question about any of that? All right, we're going to talk about the cell shape and arrangement of prokaryotes, move into cell structures, and then talk about the movement of material across membranes. We'll be talking about it for prokaryotes, but actually the movement of material across membranes works for eukaryotes as well. Prokaryotes, cell size and shape. The average size of a prokaryote is about 1 to 2 micrometers. The range being actually a little smaller than 1. It's the smallest is 0.4 micrometers in diameter, and the largest is about 10 micrometers long. We have prokaryote cells take on 3 basic shapes. There are round cells that we call coccus. There are rod-like cells that we call bacillus. And yes, we do call those rods and bacillus interchangeably. You cannot do that for round cells or spheres and coccus. Those are not interchangeable. And then the third shape is a spiral, an S type shape. These are the 3 basic shapes of bacteria and really prokaryotic cells. Most other shapes are some derivative of these 3 basic shapes. Like a spirochete is a spiral, it's just many spirals. There are different types of spirals, and actually the Vibrio, which is a half spiral, you can call it a curved rod or a half spiral. So that one could be either. And then the spirals, the spirillum, they're a little different than the spirochetes, not only in the number of spirals, but the spirillum is a rigid cell. It has a corkscrew type movement. I'll show you that. And the spirochete has a corkscrew type movement, but it's a much more flexible cell. It's not rigid like the spirillum. Any question about any of that? Let me move on to talking about some other shapes. There are some unusual shapes of prokaryotic cells, like the Stella bacteria takes on a star shape. And that's an actual electron microscopic image of them. The Haloarcula archaea takes on it's actually more of a rectangle shape, squarish. Most bacteria are monomorphic, meaning they take on a single shape. But a few bacteria species are pleomorphic, meaning the cells take on many different shapes. Oops, went too far. So here's a pleomorphic species where the cells, that almost looks like a cockeye. That's sort of a bacillus. That's sort of like a dumbbell. That's sort of like a bent rod, very long one. The point is pleomorphic, the bacteria species take on many different shapes. Bacteria, most of them have a cell wall, but there is one family that does not have a cell wall. And that family tends to have bacteria which are pleomorphic. Any questions about that? All right. We can also talk about the cellular arrangement, also called the growth arrangement or the cellular growth arrangement. This is how cells grow and then stick together. Like for cocci, you can have singlets. That's not listed up here, but that is a cellular arrangement. One single cocci. And there we can see a singlet right there. You can also have diplococci, and that's 2 cocci stuck together. I'm not sure if that's a diplococci there or not. That's sort of what they're trying to imply. That doesn't even look very cockeyed to me, but and then they can be lining up in a row. This we call streptococci. And there's a streptococci. The cocci can also grow as tetrads. Note that the plane of division differs from the diplococci and the streptococci, where the cells only divide on one plane. With the tetrads, they divide on two planes, and that's how they form a tetrad. And there it's kind of jumbled, but there's a tetrad right there. And then if they cells split on three sides, you can form a sarxinia. It's a group of 8 cells stuck together. And then cocci can also grow in a cluster. And this we call staphylococci. Staphylococci literally means cocci that grow in a cluster like grapes. And this reminded somebody of a cluster of grapes. And so they called it staphylococci. Any question about any of that? If not, bacillus also have cellular growth arrangements, but they have fewer cellular growth arrangements than cocci. You can get a single bacillus or singlets for bacilli. You can get diplobacilli. You can get streptobacilli. And streptobacilli is when they form chains where the cells are connected on the long ends. Hopefully you can see this. If the cells are connected on the I don't know what you call it, this way, I'm going to call it connected with the short sides. We don't call them streptobacilli. We call those palisades. And I think I talked about that already. If you read Mark Twain, they talk about palisade fences. That's where you put the board up this way and then that way and that way to make the fence. And those are the only cellular growth arrangements that bacilli have. Any questions about that? This shouldn't be in this slide. A Coccobacillus is not a cellular growth arrangement. This is a shape. Come back to the shape here. If we, oops, one more. If we shorten the rod and shorten it and shorten it, and if it has rounded edges, we're eventually going to get it short enough that it no longer looks like a rod. It's going to look sort of like a cocci, sort of like these cells here. That one there in particular, but these two also. And that one, in fact, almost looks like a cocci. That one's a little bit long for a cocci. That one's really long, but not long enough to be a rod. This we call Coccobacillus, in between a cocci and a bacilli. Any question about that? So this is a shape. All right, here's a few the typical structures you can find in a prokaryotic cell. The structures shown in red are in all prokaryotic cells. All cells, meaning eukaryotes and prokaryotes, have a cytoplasm. They all have a plasma membrane. and they all have DNA, which is not shown here for whatever reason. This right there is DNA. And the nucleoid region is just wherever the DNA is floating around in the cytoplasm in a prokaryotic cell, meaning there is no distinction between the nucleoid region and the cytoplasm, because when the DNA moves from one region to another, it can, and then that would be the nucleoid region. The only difference between the nucleoid region and the cytoplasm, besides where the DNA is, the only difference is they do stain a little differently under certain stains, like methylene blue. The nucleoid region will stain very differently than the cytoplasm. All prokaryotes also have ribosomes. In fact, eukaryotes have ribosomes, but their ribosomes are different than prokaryote ribosomes. The other regions shown here, or the other cell structures shown here, are not seen on all cells, like the pilus, like the sex pilus, the capsule. The cell wall is apparent on most bacteria, but not all bacteria have a cell wall. The plasma membrane should be in red. It is there. The fimbriae is not on all bacteria. Flagella, not on all bacteria. Plasmids, and the capsule, which I think I've already talked about. All right, any questions about the different structures? If not, let's start talking about them. The first structure we're going to talk about is the glycocalyx. It's a structure secreted by many, but not all prokaryotes. It's outside the cell wall. Because it's outside the cell wall, we say is that it is external to the cell. The glycocalyx is usually sticky, and there's 3 different glycocalyxes, 3 types. There's the capsule, where the glycocalyx is neatly organized. It has more molecules in it other than just water. Okay, so it has some structure to it. The capsule can help prevent phagocytosis. So the capsule can give the cell pathogenic properties, like Klebsiella pneumoniae is difficult for the body to fight off because it has a capsule. And so the phagocytes cannot ingest these cells and then kill them. The second type of a glycocalyx is the slime layer. It's much less organized than the capsule. It's much looser attached as well, and it's also mostly water. So the slime layer cannot be seen with the light microscope. To see the slime layer, you've got to use an electron microscope. And then the third type of a glycocalyx is only found in biofilms. We call it the extracellular polysaccharide. It's a material secreted by cells to make more than just a capsule around the cell. It actually forms a biofilm around the cell and all cells within the biofilm. The capsule normally is around the cell that secreted it, and that's all. The biofilm, on the other hand, will surround all bacteria in the region, even bacteria of different species. Any questions about the 3 types of glycocalyx? Remember, the capsule is the one you can see under the light microscope. It's organized and it can prevent phagocytosis. The slime layer you cannot see under the light microscope, and it's much less organized. Flagella is the next cell structure we want to talk about. Bacteria have this cell structure if they can swim or locomote, meaning the flagella is used for locomotion. The flagellum moves like a whip, and that propels the cell forward. The flagella is made of chains of protein, which we call flagellin. The flagella is mostly outside the cell. However, there is a region attached to the cell wall. And if it goes in even further, there's a region of the flagella which is attached to the cell membrane. Any question about any of that? You can find 4 different flagellar arrangements in cells, and I'm not going to go through that. It was a question in our last lab. If you got it wrong, there are 4 different flagellar arrangements. Whether the flagellum is one and only on one side, monotrichous. If the flagella is on both sides of the cell, amphitrichous. If the flagella is on both sides of the cell and there's more than one on both sides of the cell, we call that lophotrichous. And if the flagella are everywhere on the cell, we call that peritrichous. Any question about any of that? All right, if the cells can move and they're prokaryotes, then they have at least one flagellum. And I didn't mention that, so let me go ahead and mention this now. If you have one flagellum on a cell, the correct word to use is flagellum because it's singular, meaning UM is what it ends with. If you have more than one flagella on the cell, we then call it the flagella. Okay, so this cell has flagella because there's more than one. Any question about that? And we're only talking about one flagellum right here. That's why that one is called the flagellum, because altogether these are flagella. Usually when bacteria are putting down flagella, you will see the cells in different stages of flagellar arrangement, meaning before the cell can become peritrichous, it has to be lophotrichous. And before it's lophotrichous, it has to be amphitrichous. And before it's amphitrichous, it'll be monotrichous. And what is it before it's monotrichous? Come on, somebody's got to have a guess. Before it's monotrichous, the cell has no flagella.
Bacillus,
Say again?
bacillus,
Yeah, flagella less. So if the cells swim or rotate, making it appear that they run or tumble, meaning it can move like this, and really it's swimming like that and moving this way. Or the cells can sort of tumble over each other. And maybe that would be better looking at it like this. If it swims or moves, then the cell has flagella, or at least one flagellum. If the cell has flagellum, that allows the cell to undergo taxis. Taxis is the movement of a cell toward or away from stimuli. So here we have cells with flagellum. Let me blow this up. And they're responding to a light source up here, and they're moving that way, away from the light. That we call phototaxis, meaning the cells are engaging in taxis, movement away from the stimuli. And taxis could be towards the stimuli. This must not be a photosynthetic cell because it's moving away from the light. You can also have cells move towards a chemotaxic source. Chemotaxis is where the cell makes its own glucose and ATP itself. And how it does that is it strips electrons from some atom that it wants to metabolize. And then it uses that electron to make food or ATP for the cell. And these we call the chemosynthetic bacteria. And in chemotaxis, meaning that they're responding to a chemical signal, like there could be lead right here, and the bacteria know that, and it's trying to move away from the lead. Any questions about taxis? Remember, there's two types: chemotaxis, movement in response to a chemical, and that movement could be towards the chemical or away from the chemical. Phototaxis is similar. It's the movement of material, the movement of cells towards or away from light, why it's called phototaxis. And it can use that light to engage in photosynthesis, where the cell makes its own food and ATP from sunlight. And we'll talk a little bit more about that in a later lesson. Any questions on anything so far? The next cell structure I want to talk about are a special flagella or flagellum that we call an axial filament. And to tell you the truth, I don't use this word. I tend to use the word endoflagella. The endoflagella are only seen in spirochetes. The endoflagella is anchored at one side of the cell. And then it runs around the cell, shown here. Here too, but it's hard to see where the endoflagella is. And you'll note that the endoflagella is not surrounding the cell wall. Oops, too far. The endoflagella is actually inside the cell wall, meaning here we have the cell, and then the light pink is the, and the purple is the cell wall. And the endoflagella is inside the cell wall. Any question about any of that? The endoflagella doesn't beak the way a whip moves. The endoflagella causes a corkscrew type movement. Let me see if I can get this to work from YouTube. Oops, didn't get all of that. While that's loading, let me go ahead and continue talking. Remember, only the spirochetes have the endoflagella. They do move if they have an endoflagella, but their movement is different. And it moves this way and that way. And look how flexible this is. That cell was moving this way and that way, and then it just bent direction, and now it's moving that way and that way. And to move one way, it just moves in the same direction. To move the other way, it just rotates the cell moving in the other direction. Any question about any of that? Our next cell structure we need to talk about are fimbriae. They are shorter and thinner structures than flagella. There typically are many fimbriae on a cell, and flagella aren't as many, although there can be a lot of flagella on a cell. It's not as many as a fimbriae. And there may only be one flagellum on a cell. I've never seen a cell having only one fimbriae. The fimbriae are mainly used for attachment to surfaces, like this bacteria in purple can attach to the surface here with the fimbriae. The fimbriae are not used in swimming, so they don't cause the cell to move by swimming. The pili, also called the sex pili, is longer than the fimbriae. So actually on this cell here, we can see, let me see if I can blow that up even a little further. We can see that there are fimbriae here, and they're a light blue color. And then there's a pili right here. That pili comes out of this cell and literally skewers this cell. And we'll talk more about it later, but that pili can then act as a winch to bring these two cells closer together. So the pili or sex pili are longer than fimbriae, and then the pili are used to transfer DNA from one cell, meaning this cell, into that cell. Any question about that? We'll talk about the sex pili in chapter 8 a little bit more. All right, if there's no questions, let's talk about the cell wall in a little more detail. The cell wall is a complex structure. It's semi-rigid, and the cell wall is the reason for the prokaryotic cell having its cell shape. That's also true for plant cells, which have a cell wall. They get their shape from their cell wall. The cell wall is important to protecting the cell and preventing osmotic lysis. If the prokaryotic cell were to not have a cell wall, sort of like an animal cell, and then you were to put it in an environment where there's more water outside the cell than water inside the cell, which actually is most environments that a pathogen will find. Meaning when a bacteria is in our body, most of the time the bacteria will be finding more water outside the cell than inside the cell. And there's many environments like that. Like if the cell falls into a bucket of water, if the cell doesn't have a cell wall, that water will just continue to move into the cell, causing the cytoplasm and the cell membrane to expand. And the more water that comes into the cell, the more the cell membrane will expand. It'll expand up into the point where the cell membrane bursts from the cell having too much water in it. And you might have seen this in 160, and that is where you put red blood cells in distilled water. The water will move into the cell will expand and puff out, and it'll expand further until the cell membrane bursts, and then the cytoplasm will leak out of the cell, and that'll kill the cell. That we call osmotic lysis. So the cell wall protects the cell from osmotic lysis. The cell wall is located outside of the cell membrane, but the cell wall is considered the last part of the cell, meaning the most outermost part of the cell. Anything beyond the cell wall, we call it as external to the cell. And that's a little bit strange that we define it that way, because the cell membrane is the gatekeeper that determines what comes into and out of the cell. But the cell membrane is not the boundary of the cell. The cell wall is, at least of bacteria. And that's actually true of eukaryote cells if they have a cell wall. The bacteria cell wall is mostly made of a peptidoglycan. Let's talk a little bit more about peptidoglycan. Peptidoglycan is a polymer of two repeating disaccharides. Blow this up. The two repeating disaccharides that make up peptidoglycan are NAG and NAM. And then they just repeat, NAM, NAG, NAM, NAG, NAM, NAG. So the disaccharides link up in a chain forming a lattice, and then there will be one chain next to another chain, and then there will be one chain underneath. And if there's more cell wall outside of this chain, there will be a chain above this chain here as well. NAG stands for N-acetylglucosamine, and NAM stands for N-acetylmuramic acid. And these names are probably mean as much to you as NAG and NAM. A chemist would actually like these names, but either way, you can know it as NAG or NAM. The point is, peptidoglycan is mostly made up of NAG and NAM. However, peptidoglycan does have disaccharides linked. I mean, it does have the disaccharides are linked by polypeptides. So this NAG and NAM chain is hooked to this NAG and NAM chain by this polypeptide. And yeah, that polypeptide is hooked to a polypeptide here and here. But the point is, this one is linked now to that one. And then this one is also linked to that one by this polypeptide. And it's not shown very well, but this NAG and NAM chain is linked up to that NAG and NAM chain by a polypeptide going down. And the polypeptides link to each other. This polypeptide is a short chain of amino acids. And it just holds or helps hold the polypeptides together, the NAG and the NAM. Remember, the NAG and NAM form a chain. The chains are right next to each other, and then the chains are line up, and there will be a layer underneath this layer of NAG and NAM chains. Any question about any of that? Penicillin actually kills cells by interfering with the cell wall. Penicillin inhibits the peptidoglycans from linking together and forming a lattice holding the peptidoglycan together. So the penicillin could come in right here and then prevent this this polypeptide from forming. That creates a hole in the cell wall, and then the cell membrane can protrude out of that hole, so that the cell wall is not protecting the cell membrane from osmotic lysis. And that's how penicillin actually kills cells, by osmotic lysis. Any question about any of that? So here we see the gram-positive bacteria cell wall. Remember that the cell wall is very thick. It's mostly made up of peptidoglycan, the yellow layer being the cell membrane. And here we're looking at it here. It's only showing 3 layers of peptidoglycan, but in reality, the gram-positive cell has many, many layers of peptidoglycan. And I don't know how many, but let's say at least 100 layers of peptidoglycan. So it's much more than the 3 shown here. You can see that the gram-positive cell wall does have a few other molecules besides the peptidoglycan, which is actually NAG and NAM and then a short peptide. The gram-positive cell wall has teichoic acid, and that runs through the peptidoglycan, helping to hold the peptidoglycan together. You can also find lipoteichoic acid, and lipoteichoic acid is teichoic acid that has a lipid attached to it. The lipoteichoic acid runs through the peptidoglycan, but it also runs through and attaches to the cell membrane. Any questions about any of that? I think I already talked about that. Teichoic acid and lipoteichoic acid may help the regulation of the movement of cations, meaning a positively charged ion can move or can move towards the cell or into the cell by running down the teichoic acid in the cell wall. However, the cations and other molecules do not need the teichoic acid, meaning anything that can dissolve in water can get right through the cell wall, at least the gram-positive cell wall. So anything that dissolves in water could go right through the cell wall. If the molecule does not enter the cell, it's because the molecule cannot get through the cell membrane. The polysaccharides on the gram-positive cell can provide antigenic differences that we call antigenic variation. And actually, molecular biologists have used the antigens of bacteria species to make or to define the strains of a species. Remember, the strain is the same thing as a human race. We have at least 3 human races: blacks, Caucasians, and then Asians. The point is, there are different races and there's different strains to a bacteria species. And that strain can be defined by the antigenic variation of the cells. And that antigenic variation comes from the polysaccharides in the peptidoglycan. Any question about any of that? All right, if not, let's quickly talk about the gram-negative bacterial cell wall. It is similar to the gram-positive, meaning it has a cell membrane, which is not part of the cell wall, and then it has a peptidoglycan layer shown in the thin purple, which is shown here. And then outside of the peptidoglycan, we have a lipopolysaccharide layer that actually forms a bilayer that we call the outer membrane of the cell wall. Any question about any of that? All right, to point out the differences, remember the peptidoglycan for the gram-negative cell is much thinner than for a gram-positive cell, bacteria I'm talking about. But the peptidoglycan layer is more than one layer shown here. For the gram-negative cell, we have an outer membrane of the cell wall. And when we look at that outer membrane, this layer of the outer membrane of the cell wall looks the same as the cell membrane. That is largely because it is pretty much the same. The main molecule in both the cell membrane and in the under layer of the bilayer, meaning this layer of the bilayer, is actually phospholipid. So that's the main molecule. You do find phospholipid in the top layer of the outer membrane of the cell wall. So it's all of this area here, the little yellow and the phospholipid tail there. But we do find other lipids in the outer layer of the bilayer. And that's that blue lipid A here. It's actually lipid A. Attached to lipid A, we have an O polysaccharide, and that creates the hair-like structure on the lipid A. Together, the lipid A and the O polysaccharide make up the lipopolysaccharide. And that's different. You don't have lipid A in the cell membrane or even in the lower layer of the outer membrane of the cell wall. Lipid A is important because lipid A is an endotoxin. So lipid A makes this cell toxic to our cells because lipid A is an endotoxin. Everybody got that? So if a cell is gram-negative and it has a cell wall, it has lipid A in that cell wall, and lipid A is an endotoxin. So this infection is, well, a little more complicated and potentially more devastating than a gram-positive infection, assuming the gram-positive infection doesn't have a toxin in it. Any question about any of that? All right, we have a few other molecules in the cell wall of a gram-negative cell. We have the porin protein, and the porin protein makes a channel protein that allows molecules to leave or enter the outer membrane of the cell wall. Meaning if the molecule wants to move through the outer membrane of the cell wall or move into the cell by moving into the outer membrane of the cell wall, that molecule, unless it is soluble in the lipid bilayer of the outer membrane, that molecule has to move through the porin protein. The porin protein is similar to a transport protein in the cell membrane. So if the molecule, like an amino acid, is one that the cell wants, the cell will make a transport protein and put it in the cell membrane. And then that transport protein will move or pump that amino acid into the cell. If the cell wants the amino acids, and all cells will, then that amino acid has to move through the porin protein. The difference between the porin protein and the transport protein is the transport protein obviously is found in the cell membrane, the porin protein in the outer membrane. The transport protein generally only transports one molecule either into or out of the cell. So we'll have different transport proteins for different molecules. The porin protein transports many molecules through the porin protein. If the cell wants it, then most likely that molecule will come through the outer membrane through the porin protein. If the cell doesn't want it and it can block it, like penicillin G cannot get through the porin protein, and then penicillin G cannot damage the peptidoglycan of the gram-negative cell. All right, any question about porin proteins and transport proteins? Or lipoprotein, we can have a lipoprotein, and this is a protein in the cell wall that has a lipid attached to it, and that lipid portion of the protein will attach either to the outer membrane of the cell wall or to the cell membrane, or both, in which case you'd have to have a lipid on both sides of the protein. All right, any question about that? Remember, the main molecule of both the cell membrane and the outer membrane of the cell wall is a phospholipid. But lipid A is also found in the outer membrane of a gram-negative cell. And lipid A is an endotoxin. Here we're looking at the gram-negative cell in a little greater detail. I've talked about lipid A and the O polysaccharide. I mentioned that the cell wall has a peptidoglycan layer and an outer membrane, and together they make up the cell wall. There's also a periplasm, which is part of the cell wall. I don't use the term periplasm. I already talked about lipoprotein, phospholipid. Okay, I think I've talked about everything. Any questions? If there's no questions, I think I'll stop here. Yeah, because I won't be able to finish talking about the gram-negative and gram-positive cell walls. So we'll stop here. And I'll see you at 6:30 for the lab.